Hello,
Okay,
I
good.
am Michelle Shiver. I am Director of Research for Star Roses and Plants. I'd like to welcome you.
It depends on the seat lot because I think.
Oops.
Okay, let's start that over again. So I'm Michelle Shiver. I'm Director of Research for Star Roses and Plants. I'd like to welcome you today to our webinar on managing rose rosette disease in the landscape. We've got a set of panelists from around the country who are experts in this area. They're going to be discussing with you today diagnosis, prevention, and treatment of rose rosette in the landscape. Following the formal presentation, there will be a question and answer session. To submit questions, please type those in the question dialog box, and then we will answer those at the conclusion of the formal presentation. And for anyone who is not available to see the seminar today, it will be recorded and posted on the Star Roses and Plants website. With that, I'm going to turn it over to Dr. Tom Evans from University of Delaware, and he's going to get us started.
Thank you, Michelle. Today, we're going to give you an update on the research progress and the challenges we've encountered during our study of rose rosette disease over the last now 3.5 years. So you'll be hearing lots of acronyms, but obviously you're all familiar with RRD. David Byrne and myself have been doing the introduction for this, and then I'll be turning it over to others to finish up the presentation. Many of you probably know that RRD was first reported in the early 1940s. Remember, this is all done by symptoms alone, but at least symptoms that are consistent with what we know as RRD today were first reported in California, Wyoming, reports from Manitoba and Western US and Canada. And then that was later found first in Rosa woodsii, a native rose, and then later on cultivated roses later on. The march of RRD over nearly 45 years or so took place from its first origins in California in the western United States and Canada, all the way across the United States. And where you see all the green on this map, that's not RRD. That's, in fact, our native populations of what is an invasive foreign rose, Rosa multiflora. And as we'll talk about later, has contributed to this march from the dry west all the way to the east coast, finally arriving on the east coast of the United States in about 1996, where we reported it in Delaware. Since that time of its spread, it spread, as I said, hang on, this is jumping now, I got to go back. RRD has been causing considerable loss to the rose industry due to infection in the Midwest and eastern regions of the country. Part of this movement appears to have been possible primarily because of the large areas of multiflora rose, particularly east of the Rockies and east of the Mississippi, where you see all the green on the map. That rose is not going anywhere. It's naturalized in the United States and serves as a reservoir both for the mite and for the virus. There are 3 players in this particular epidemic that's been occurring in the United States. And the 3 players are first the pathogen, which while we always suspected it was a virus, was not characterized or identified fully until 2011. And now we know it as a new member of the Amara virus group, and we call it by the acronym RRV, our rose rosette virus. Once the virus is in a plant, it's vectored there, by the way, by a very small areophyte mite. It makes its way through the plant and eventually kills it. The very small vector of this, the small areophyte mite, harbors the virus and then can transmit it to the plants. The mite itself moves quite easily from plant to plant, short distances, by walking longer distances on the wind or by human help. The mite can also move long distances as it floats on the wind. These mites can balloon, as we call it, on the wind and travel great distances. The final player in this, as we talked about on the far right there, is multiflora rose, which is naturalized throughout the Midwest and eastern U.S. and serves as a reservoir for populations of the vector and the virus which it harbors. When this exposes cultivated roses to the vector virus and therefore the disease. As a result of the ensuing epidemics in RRD and in cultivated rose, this group, led by Dave Byrne, had a large rose summit a number of years ago, sponsored by the rose industry and organized by Star Rose here in Newark, Delaware, where most of the participants on this grant met, along with many others, to develop a grant concept and ultimately produce a proposal which was entitled Combating Rosette Disease. This was subsequently submitted to USDA and was funded in the first round, which is an unusual event for most people who are looking for grants and shows the importance of this disease to the rose industry and the shrub industry as a whole. This proposal had 6 components. Today we're going to focus on the first 4 of those. One of them was to develop better diagnostics, approaches that would make for quicker and more accurate testing of the pathogen, to further study the virus and the mite under field conditions and develop best management practices for managing the vector and therefore the virus, to search roses for resistance and develop quicker methods to incorporate this resistance into new rose cultivars, and lastly, to monitor the disease and its spread, because as long as we have RRD and we have multiflora rose, then we still will see continuing spread of the mite and its pathogen, the virus. The group that's here today and participates on the grant as a whole is spread out across much of the eastern United States, from people on this call like myself to people at USDA in Beltsville and elsewhere, University of Florida, AgriLife in Texas A&M, Oklahoma State, where Jen is based. We have people also at University of Tennessee. And so for all of the work, we are spread a long ways apart, but that's needed for some of the things that we're doing. I'd like to also acknowledge that this has support from the American rose industry as well. Don't want to miss on that one. The next map shows the locations of all the people I just mentioned and many that I didn't. These are collaborators, breeders, monitoring network, some that are the PIs on the grant, others that are collaborators and that have made the work possible, spanning something like 31 of 48 of the continental U.S. states. Without all those people, this work would not be possible, and it's a good group to work with to get this kind of project done. The biggest challenge for the group is not only identifying resistance, but then incorporating that resistance into rose germplasm that's acceptable to the industry. So the breeding is the ultimate goal. Breeding of resistance to RRD is the ultimate goal, and how to speed up that breeding secondarily. This work is being led by our project director, Dave Byrne at Texas A&M. It's a long-term aspect of this project. The development of RRD-resistant roses has 2 principal parts. First, finding sources of that resistance through screening work that's being done at several locations in the project. And then once finding germplasm that has the resistance, incorporating that into cultivars that are commercially acceptable. This will take a number of years, and for that reason, Dave Byrne has been working diligently to find methods for speeding up the breeding process. So we're looking for ways to accelerate that breeding process. Thus far, we know there is resistance to RRD. We have several lines, accessions, and cultivars even that show resistance to RRD, but we are now confirming that. It's going to take, we've looked at some 900 individuals, 900 genotypes, out of which 850 are obviously susceptible. That leaves us with about 50 that we're still hopeful that there is some source of real genetic resistance in those, and they're not just escapes. Those 50 are being further examined to determine if they truly are resistant in replicated trials here at Delaware, in Tennessee with Mark Wyndham, with Jen Olson in Oklahoma, and in Texas with Kevin Ong and others. I will say many of us are confident that there is resistance out there someplace. So I think the breeders are more confident than the plant pathologists, but we do know if we look hard enough, we will find resistance. And so these replicated trials that we're performing are an endeavor to confirm that in fact we do have resistance in germplasm that can be more easily moved over into acceptable rose cultivars. There are some rose species that appear resistant, Palestris and Satigera, as well as a few other cultivated roses. And from the breeding perspective, though, it can take many decades to incorporate this into the right material for release. So how do we make this quicker? How do we do this? We use what's called DNA-informed breeding or marker-assisted breeding, but using current-day genomics and sequencing. It's now common within many plant species and crops to use DNA-informed breeding, which will greatly improve the speed at which we can move resistant material to the field. Consequently, Texas A&M is working with these molecular genetics approaches to develop such a pipeline to bring this material to the industry quickly. This should essentially cut the time in half by some estimates of bringing material that is acceptable to the industry. So right now, we have been working on this project for almost 3.5 years. We've learned a great deal. And what follows will be talks to discuss our current options for disease management, starting with Kevin Ong on diagnostics and RRD monitoring aspect of the work. With that, I'll turn this over to Kevin Ong at Texas A&M.
Thank you. What I'm going to try to do today is to show you some of the symptoms that we see with Rosette and give you some of our observations and our speculations on what we see. I also want to share with you the type of testing we have currently available for doing confirmatory diagnostics. So let's move on. All right, so this is typically what you would see on a rose, that it's dying. Well, the trouble with this image is it's hard to tell whether it's Rosette or not, and unfortunately this is one that actually succumbed in part to Rosette. Now let me show you some more details in terms of symptomology. A common symptom that we tend to find is long, thick shoots and stems. If you were to walk towards a bush, typically you will see 1 or 2 shoots that seems to extend beyond that shrub, and very often this branches or canes appear thicker. On some cultivars, red growth is very prominent, so you have excessive reddishness. Now, keep in mind that when we say excessive reddishness, we refer to the cultivar. If you see a lot of red in a cultivar that's not supposed to be red, well, that's a good sign that it could be Rosette. Witch's broom, a real common example, or at least a symptom that people recognize very often. So if you think about a witch's broom, what happens is you have the leaves being formed, but that you have abnormal leaves, and then the internodes does not elongate properly, and what you end up is a real bunchy group of leaves, or in this case flowers. So rosetting is a common symptom that we see on a lot of Rosette samples. Excessive thorns, and here qualify again for that particular variety. In this image, not only do you see the thickened canes, but you also see the excessive thorniness. What's interesting is for the most of our observations, we notice that the thorns tend to be somewhat hard rubbery. So it's not like a woody type solid, but it's more like the hard rubbery type thorns, and it can still hurt you. But keep in mind, again, when we talk about excessive thorns, this is on cultivars that if you see thorns on a cultivar that's supposed to have little or no thorns, then start wondering if it might be Rosette. Yellowing and stunting is another one of those symptoms that we tend to see on some of the newer plantings, and this is usually associated with abnormal leaf formation, occasionally rosetting, and on some larger bushes, what's will be interesting is you might see 1 or 2 canes exhibiting this sort of symptom, but not the whole shrub. So there's some variation, but yellowing and stunting could be one of those symptoms that is seen with rose rosette disease plants. Leaf distortion. Um, in this image at the foreground, we not only want to show what leaf distortion looks like, but also we tend to see some of the reddening or anthocyanin build up in the leaves. The leaves may be curled. Very often, the leaves is thin and strapped. Um, and here, even though we're trying to show a flower discoloration or distortion, but if you look at the leaves on either side of it, that's that excessive reddishness, and the leaves is abnormal. So keep that in mind. Those are some of the symptoms is associated with rose rosette. Now, what's interesting about this picture too, it almost looked like some part of that flower is reversed, and you're seeing sort of leafy-like tissue coming out of that flower. That's another um, uh, some of the funky things that could be associated with rose rosette. Branch and shoot dieback. Now, what we tend to find too, if you have rosetting, if you have the yellowing that progress to excessive reddishness, as it progress along, very often we see branch or shoot dieback, but it's really hard to tell if it's just branch or shoot dieback and attribute it to rose rosette, because there are several other disease pathogens that can cause such symptoms. So in all, the thing to keep in mind is you may have rose rosette disease even if you only see one of those symptoms. And that's one of those interesting things that we're still trying to figure out. Several years ago, I had an undergraduate student trying to catalog the symptoms that we see and try and correlate it to the varieties or cultivars that we find, and we could not match up anything. So the symptoms that we find are quite variable even within a particular cultivar. So quite often, if you see a rosette and you see several of the symptoms that I showed here, that's a very good chance that it is rose rosette. But even if you just see one, it still could be rose rosette. Makes it a little bit harder to diagnose visually. Now, in terms of diagnosis, in a diagnostic clinic, so what we're trying to do is test for the presence of the virus. Well, in terms of a virus, there are two common ways that we are able to detect viruses in general. That's the ELISA, or enzyme link immunosorbent assay type methods. And here I showed an example of a lateral flow device similar to a pregnancy test type stick. Just to be clear, we don't have this tool yet for rose rosette. A couple of folks in the research group are working on that. We hope to have that real soon, sometime this year. But at this point, we do not have a lateral flow device detection tool that's available on the market. So what we do rely on right now is polymerase chain reaction methods. PCR methods basically take advantage of what we know about that virus, and we target specific areas that are unique to the virus and use this tool called PCR that basically um, amplifies the gene of interest, and so we can actually tell whether that virus is present or absent. And so currently the PCR methods is the method that is used, or at least the approach that's used by various diagnostic lab to detect for rose rosette virus. Now, I do want to point you guys to a website, roserosette.org. So one of the tests that I've been given is to help with the monitoring of rose rosette disease and trying to capture information on where we find rose rosette and on what cultivars. All this effort is trying to help our breeders to see if we can identify cultivars that may have a resistance or tolerance to rose rosette. So roserosette.org is a website which we hope serves as the clearinghouse and provides some of the information from the various states in terms of fact sheets. It's made available on this website, but it also has a reporting module. where if you do see Rosette, or what you suspect is Rosette, you can actually click and report that, and we ask you to send a picture, and I'll tell you how all this works. So on this slide, you actually see a tab there that says reporting, and if you do click on that, and by the way, this website is optimized for use on like phones, smartphones, and tablets. It'll work on a desktop or laptop, but it works really well on the phone, which is what we were trying to do so folks can take pictures on their phone. So if you click on reporting, you'll get a page that looks something like this. It will ask you for the cultivar, the host. So there's a drop-down list, and we try and populate this list with as many cultivars that we know that are available in the market today. And if it's not, then there is a place where you can put in as rose species, and then we may reach out to find particular um, uh, variety that you may be reporting. There are required information that is needed to be entered, which is the state and the county that you're in. Um, we do ask for latitude and longitude if you have it, but this is not required information. All that information is just trying to be able to at least get some resolution as to where Rosette might be or is. Um, on the smartphone or on the tablet or even on the desktop, there is this uh, entry fields at the bottom which talks about images. So you can upload images onto this reporting. At this point, we actually limit up to 4 photos per report. So we ask that when you report on an individual plant, and you submit up to 4 photos of that individual plant of the symptoms that you see. So if you got 2 or 3 different plants, you actually enter it 3 or 4 times. This way we can help to keep track of individual plants and the individual cultivars the data that or information that we might get. It will ask for your contact information, first name, last name, and also there is a check there says I am not a robot, because we don't want somebody to put this on a robot and spam us. But what we want to do is hopefully utilize and basically encourage the general public to come help us gain more information about this disease and on what cultivars it get on. When you're done, you click the submit report, and that entry will go to the web page server. Now, this is what happens when you submit a report. When you hit submit, that report will actually they'll look at which county or which state, and then a verifier will be basically someone who had volunteered, usually a plant pathologist that or a trained verifier. What happens is they will get an email and said, we got a report, and you said that you're going to be able to check on this report. So what they'll do is they'll actually go in, look at those images, look at the information that's provided, and they make a choice here. They either say, yes, we already have seen this on this particular variety. It looks really common for Rosette on this particular variety in this area. And they verify it and said, yes, confirm, true visual. But if it's on a new variety that we don't have any information of, they may basically make the software says, and send an email out and said, we're interested in this sample, and we would like for you to submit a sample to a diagnostic clinic. And if I believe that there will also be a packing slip that comes with this email to help to make that submission easy. Now, if there's a situation where the image is blurry and all, but it's in some place that is exciting for us, we might say, hey, we need new photos.
We handle my registration stuff perfectly in my credit cards, and I thank you for this is for ICPP 2018, obviously. But I also registered my students.
Anyway, sorry about that. Somebody might want to mute his mic there, Tom's mic. Okay, where was I? So If you submit the sample in and um, and everything goes well at the clinic, and we do the testing, the confirmatory testing, you will receive a follow-up report, and that report will say, yes, there is Rosette virus is detected, therefore what you're seeing is in fact what we believe to be Rosette disease, or no, there is no Rosette virus detected. So what you're seeing that you think is Rosette disease symptoms might be caused by something different. There are quite a number of mimics, and if you want to get that information, go to roserosette.org, and there should be some information there about the mimics of Rose Rosette. And what you get to do is what we hope is you get to be a part of the folks that help us to get closer to a solution, and we hope that this actually speeds up some of the information that we have on resistant and tolerant varieties, so at least narrow it down. Now, I'm going to turn this over now to Jen Olson. Thanks.
All right, thank you so much. Today I'm going to talk to you about best practices to prevent rose rosette disease in the landscape. So when we're talking about managing this disease, there's a couple of things to keep in mind. The first thing is that rose rosette is a complicated disease. This is a disease, it's a virus that is carried by a mite that feeds on the plants. The mite is microscopic and it's windborne. And so because it's a complicated disease, then one single management strategy is not going to be adequate at providing good control. So we're going to need to use an integrated pest management or IPM strategy. What I've put together for you is a compilation of best management practices for rose rosette disease. One reminder is that rose rosette is not nationwide. In some areas of the United States, it's absent. And so depending on your location, it may affect how you are managing this disease. Even in some cities, we can find areas of a city that have rose rosette and then other areas where it's absent. So if you are in an area where rose rosette is present, it just may take some additional care and planning. One of the first and most critical steps of managing rose rosette is to eliminate the source of the virus. So this virus is out there, and the two main places that we find it are, number one, in landscape plantings that are unmanaged. So if we can identify these and remove these plants showing symptoms, as you see in that upper image, that's one way to reduce the level of disease in an area. It's also likely in many areas of especially the eastern and central United States, multiflora rose and other wild roses commonly harbor rose rosette. And so they harbor both the virus and the mites. And so if you have plants like this one shown in the lower image growing on a roadside, in a wood line, or just a wild area, by removing these plants, that helps cut the amount of virus that you have blowing into the area with those mites. So very commonly, if we eliminate these as a source, that's one way to reduce the level of disease. Sometimes we have had the question about, well, if I have a rose with rose rosette, is it possible just to save that rose by pruning the disease out? And unfortunately, Dr. Mark Wyndham and his colleagues at the University of Tennessee have done several research trials looking at this. And what they have found is that pruning disease shoots is inconsistently effective and it's not recommended. It just doesn't work that well. And sometimes we have people say, oh, I saved it because I didn't see the symptoms again until the next year. Well, it may be a delay of up to a year before the symptoms express. The virus can be in the roots or the crown of the plant, and so there just may be a delayed or a latent period before it re-expresses itself. So in this image, you can see this was a failed attempt at pruning where the arrow is pointing as a pruning cut, and we see when one of those lower buds broke, the shoot that came out is showing the symptoms associated with this disease. Another thing about pruning is quite often those microscopic mites have already moved on to other parts of the plant. And so just by cutting that out, you may not have addressed the problem. So if we do find this disease, it is suggested, rather than trying to prune, to actually remove the symptomatic plant. And so you want to remove the entire plant. It is helpful if you can cover it with a large trash bag or wrap some plastic sheeting around it, cut it at the soil line, and discard it in the trash. It's also suggested to remove the root ball. Following pruning, if you have other roses in the immediate area, you may want to use an application of a miticide to treat those healthy plants in case we disturbed any of the mites in the process of removal. And we'll talk more about miticides shortly. When you do remove a plant, it is important to revisit that area to check for what we call suckers. So if you've left any larger roots behind, some rose cultivars are more likely to do this than others, but they may produce a sucker from that root piece. So the circled plant in the image is one of these suckers that is coming up from a removed plant that is showing symptoms of rose rosette. So be sure to rescout the area before you replant. It is possible to replant in the same site. Although you could do it the same day, you do need to come back and check for these suckers, but usually it's best if you can wait a couple of weeks to allow time to tell if those roots are actually gone. At this time of the year, much of the country still has most of their roses in dormancy, and so if you have not done dormant pruning, just some tips for you. We have found that the primary site where the mites survive the winter is in the association with old blooms and leaves. So when you are doing that dormant pruning, be sure to remove those old flowers, those spent blooms that are still hanging on the plant, and discard them in the trash. This is just a cultural method that will allow you to reduce the mite population. Following the pruning, be sure to clean up the debris. It is never suggested to use leaf blowers around roses unless they are 100% dormant. So it is much better if you can remove clippings by hand, but be sure to discard them in the trash. Some of those mites might be on that debris, and they could simply crawl or move back up to the rose plant nearby. Following pruning, it is helpful for many reasons to make an application of a dormant oil. So there's many different types of oils available. Often mineral oil or neem oil are the active ingredients. They're petroleum products that we spray on these plants, and it's basically smothering those any mites that might remain on the plants. Even if it's not rose rosette, an application of dormant oil can be very helpful for many pest and disease issues at reducing the likelihood of a problem in the spring. Once we get into the growing season, just some other tips to keep in mind. First of all is that healthy roses are less likely to develop pest and disease issues. So ensure that your roses are properly planted and receiving adequate care throughout the season. So the rose in the image that we see here is showing symptoms of a nutritional or perhaps pH problem. So if we can improve the overall health of the plant, overall it's going to be healthier and more likely to resist pest and disease issues. Throughout the season, deadheading is quite beneficial. Those mites are often in association with the open or spent blooms. So throwing those away, discarding them in the trash, is another great way to reduce mite populations through the season. It's never suggested to use leaf blowers around roses during the growing season. We know that the mites that carry this virus are wind-borne. And so if we're introducing artificial wind, we're more likely to spread them more often and to longer distances. During your workday, should you encounter a plant that has rose rosette, it's not a good idea to visit other roses that day. Although we know that this virus is not transmitted by sap, it could be possible that if you have debris on your clothing, on your equipment, your gloves, and you're moving to another site with healthy plants, you could be carrying some of those mites with you. So if you have a problem spot where you've been fighting rose rosette, try to schedule visiting that landscape as late in the day as possible. Regarding new or replacement installations, it is possible to replant a rose in the same site. Just remember, you do need to remove that root ball and verify that there's no suckers. When you do that, also gather up any debris, leaves, or blooms that have fallen on the ground and throw them in the trash. The image that we see here, this is a great candidate for replacement. The roses in the background in the left show symptoms of rose rosette, while those in the foreground are beautiful and healthy. So we could remove those plants in the background, replace them, and then it's a good idea to treat the roses on the property with an application of a miticide, so that if we did disturb any mites, that we will take care of them before they have the opportunity to spread the disease. When you are selecting new or replacement plants, always inspect them for many pest and disease issues. We want to start healthy and stay healthy. And although rose rosette is not very common in the nursery trade, it does occur infrequently. So just make sure that the plants you're installing are healthy. For those new plantings, be sure to use proper spacing and don't overcrowd your roses. Looking at those in the image, these roses are large rows, and these are planted in such a way that not much of those leaves overlap. Within a planting, most of the movement of the mites is plant to plant, leaf to leaf, so they just crawl down the row. And so if we can make sure we have the proper spacing so there's not a lot of overlap, it reduces the likelihood of the disease moving. In a high-density planting where the plants are spaced very closely, you're going to have a lot of leaves overlapping, and so it may require regular treatments with a miticide to prevent an infection. Dr. Wyndham has also done some research looking at other options with new installations or replacement installations. One of the things that is very helpful is if you can incorporate taller barrier plants, structures, or fences into your designs. Put them on the windward side so that if mites are blowing onto the property, they're intercepted by those structures or non-hosts, and they either starve and die or they simply blow off to another location. It's one way to reduce the likelihood of it showing up in your planting. So those roses in the foreground of this picture remain healthy. Another very helpful thing is to incorporate both roses and non-roses into your planting. So we have mixed plantings. It's a more interesting design and it helps interrupt the disease cycle. So should it appear, then you have the opportunity to remove and replace plants before you have a significant problem. If you are using a lot of roses, try to use different cultivars. What we have observationally observed is that roses vary in resistance and susceptibility to rose rosette. We're not ready to release names of varieties or anything. We're just not that far into these trials. But we do know, just using different cultivars, different genetics, that it can be very helpful in slowing the disease progress. So Dr. Mark Wyndham is not able to join us today, and so I'm going to cover some of his research that he and his team at the University of Tennessee have done regarding chemical control. So they have done extensive trials on the effectiveness of miticides at preventing rose rosette. And with their trials, they looked at 2 different trials evaluating the effectiveness of miticide applications over 2 years. So they actually installed, and these plants were maintained for 2 years. So they continuously got larger and as they did their trial. During the trial, they applied products throughout the growing season on a weekly schedule. And when Dr. Wyndham set up these trials, it was expected that all of the products would fail. And so in hindsight, maybe they didn't do it as well as we would have liked. There was no rotation of miticides, and the highest rate was applied every week. And so they put out a lot of product, and that's not something that we would recommend. However, it did give us some data on products that do seem to reduce the mite populations or keep them near zero and prevent rose rosette from developing. So in his trial, at the end of the 2-year period, 100% of their plants that were the control developed rose rosette. And by control, that meant that they were sprayed with only water. The products that I'm going to show you in the following table provided quite effective control of rose rosette. They had 0% incidence at the end of the 2 years. And so you have them in front of you. I will read the 4 trade names of products that after the 2-year period, they had 0% incidence. They were Akari, Contos, Forbid, and Taustar. Now, just to note, they did have a couple other products in the trial. One of those was Seven or Carbaryl, and it was not effective at all. It had similar results as the water only, so near 100% incidence. So Seven would not be a good product for preventing rose rosette. Also included in the trial were horticultural oil alone or Avid Plus horticultural oil. And after the first year, both of those products were performing quite well under lower disease pressure. But when they got into the second year, because there was so much infection in the trial from the control plants, that these plants were just being showered on by mites, that these 2 products, horticultural oil or Avid Plus horticultural oil, just didn't hold up. They still provided some control, but it wasn't as good as we would like. For residential landscapes, perhaps you have clients that may want to do their own treatments, not the commercial products. The products that Dr. Wyndham is suggesting, the best product would be bifenthrin. It is for the trade name, it was on the previous slide, the bottom one, Taustar, but there are homeowner versions of this that are sold as bifenthrin, and it is a systemic product, and it worked very well as a commercial product, so this is what is now being recommended for residential landscapes. It's also helpful if you can use horticultural oil. Although horticultural oil will not work under high disease pressure, under low disease pressure, it is effective. And it would be these oils, so these different products, the active ingredients might be neem oil, mineral oils, petroleum distillates. All of these products that you can find there are versions that would be acceptable for organic gardening. So at this point, Dr. Wyndham currently has trials evaluating different spray schedules and schedules that use different mixed chemistries, something more like we would hope to use in a landscape. And unfortunately, those are in progress and we don't have those results yet, but stay tuned for updates on that. So just to conclude, remember that management for rose rosette requires an integrated approach. One strategy isn't going to work, and if we can incorporate several of these different strategies, it's going to reduce the likelihood that rose rosette gets into the planting and allow us to better manage it should it appear. Rose rosette is not present nationwide, so strategies in one city may vary from the next. And roses provide such a beautiful addition to the landscape. We want you to continue to plant them and enjoy them. It's a plant that we would miss greatly. It's just going to be a little bit more challenging and more interesting to grow them. So thank you for your time.
Well, thank you to all of our speakers today for providing such wonderful information. We're now going to open it up for a question and answer session. We already have a few questions that have come in through the dialog box, but for anybody who wants to submit additional questions, please do so at this time. So we'll just start with some of the first questions that we've already received. So the first question we got was, for the reporting website, how long does it take to hear if you want a sample?
That's a great question. It depends on how quickly the verifiers get to that reporting in. Typically, we like to get to it within 1 week, but it's really variable at this point. And we are hoping to get more verifiers online. And so I think when there are more hands involved, the shorter the response period.
Thank you, Kevin. Our next question is, will RRD survive in the roots?
I'll take that too. You want to do that, Tom? Go ahead.
Sure. Well, we do know we can get the virus out of the roots. Survival meaning that, if you have active living roots, as Jim was saying, if you haven't removed an entire plant and there's enough material there to sustain a root mass and it was an infected plant, we can with our PCR test detect RRV in the roots, and we've been unable to transmit it out. That doesn't mean that the mite may not be able to if they're surface roots, but certainly roots do contain virus. Let me put it that way, of RRV.
Let me jump into that and add to it. The biggest concern that we see with roots, and we do recommend that if you pull out the plant, pull out the roots as well, because if rose rosette is in the roots, there's roses have a very bad habit of putting up suckers. And so those suckers are likely to be infected, and that's where the aerophyte mites will be able to pick up the virus. So yes, the virus can be detected in the roots, and we believe that it is in the roots on some plants.
Right.
All right, so our next question is, reporting in places not managed. What?
Reporting in places that you don't personally manage. What do you do?
Okay, so if you've got a plant that's not in a place that you're not actually managing, what do you do?
Excellent question. I don't have a great answer for you because this is not a regulated disease in a sense that, is quarantine issue and all that. Uh, I do advise our master gardeners and some of our landscapers, best thing is to approach the owner or whoever is taking care of that and hopefully educate them about it. You can suggest to them to remove those plants, but you cannot make them remove those plants. So we are trying through this effort also a big push in educating folks what rose rosette disease is, what rose rosette virus is, and so the in my mindset, Kevin Ong's mindset, is that through education, then we can have a collective effort to continue to enjoy rose into the future.
And Kevin, education even of the municipalities, the governments, because most, even city governments, often have clauses, just about like the states do, that say it's illegal to knowingly maintain a plant with a disease that's transmissible on it. And it does say that because I've had the same problem with like fire blight in our cherries. Cities and states don't do anything about that because it's not regulated, but there are usually pieces of the city code even to maintain them. So getting to the municipalities would be a good idea also.
Absolutely.
All right, so our next question is, can virus be spread by pruning, such as tools, and do you recommend disinfecting tools between each cut?
Have you guys seen any data? We have anecdotal data. I know that there's no reason why it shouldn't be, but we don't have strong data absolutely that it is. The mites are essentially mechanically transmitting. It's probably more of a probability issue than anything else. What do you think, Kevin?
Well, there are actually two old papers that did this work on multiflora roses, and so they have shown that they were not able to transmit rose rosette disease. Remember, this was before the time that we actually confirmed it as rose rosette virus, but it was suspected to be a virus, but they could not transmit it using pruning. So keep that in mind, and there have been other anecdotal evidences, and I do know that my colleague, who has since retired in Overton back in the 90s, had a student that did this work. It was never published, but they were also not able to get rosette disease to be transmitted. So they would use a pruning tool on a diseased looking plant and then go down the line to a healthy plant, and they would not get those plants uh, to get rosette disease. So at this point, based on the information we have, both the research that was done previously and the anecdotal evidence, we don't believe that pruning is a primary or a very effective method of transmission. So,
However,
however,
There are errors. Be careful.
keep in mind two things is that this presentation is focused on rosette, but it is possible to transmit other viruses and other diseases by pruning. So occasionally sanitizing your pruners is always suggested. And where we are concerned is that although the virus may not be sap transmissible, it may be possible that you may collect mites on your pruning tools or equipment and move them down the line. So if you're using hedge clippers or something, it may be possible to, carry those mites throughout that planting so that it's a method that you could possibly spread them.
And they're so small you will never see them.
That is true. All right, so our next question is, when reporting, do I need to leave the rows in place or can I go ahead and remove the rows?
In the reporting part, if you do suspect that it's rosette, I would say pull the rows up. The reporting is just a confirmatory approach. Hopefully, if we can get, that's why I like to get back as soon as we get a report in, because if we need to confirm it, we can get a sample. So if you
could, in the meantime, if you are reporting it, you could collect some of those symptomatic shoots, roll them in dry newspaper, and place them inside of a plastic bag, and just keep them in the refrigerator until you hear back. And that way, if a sample is requested, you're already ready to send it.
Excellent suggestion, Jen.
And roses will hold like that for a good week to be a good symptom, a good sample for putting to the system.
And just to clarify, is we not the whole plant, but symptomatic plant tissue. So you a few of those symptomatic branches in that newspaper in a Ziploc bag would be great. You don't have to stick the whole plant if it's a big bush in the refrigerator.
And it's always advised to try to find some open or spent blooms and include that with your sample. If you want to know if mites might be present, that's the best place for us to look under the microscope.
Great. So moving along to our next question, are there any distinct differences in symptoms between rosette and those symptoms associated with herbicide damage?
All right, I'll take a stab at that.
Red color.
that is one. Red. Yeah, that's one of it. I think one of the most common things that we have seen people mistaking glyphosate type damage for rosette. Now, what has been interesting for us at the in Texas is there are times that we see those symptoms, and we test it. Sometimes it comes up rosette, sometimes it does not. But usually, when it does not, and you ask some questions, you can always track it back to somebody using glyphosate, either in that landscape or close by. So that's only one example. There are other kinds of herbicides, hormonal type herbicides, basically can result in epinasty or abnormal growth, and sometimes those could be mistaken for rosette as well. In terms of distinct, typically we think like Jen said, the excessive reddening on that tissue is often a giveaway. It's not a foolproof symptom, but it is a giveaway.
Particularly not early in the spring, obviously.
You usually don't find the proliferation of prickles or thorns with chemical injury is another thing, and also the succulence, that the stems are often thickened. That's seldom found when there's chemical injury involved. But when in doubt, you can have it tested.
I'm not sure what that is.
Parlor.
At least it's not me this time.
All right, so in regards to testing, our next question is typical symptomology plus the presence of aerified mites observed on tissue under the microscope a robust diagnostic strategy if PCR is not available?
That is an excellent question. And I will say this, the Texas Plant Disease Diagnostic Lab prior to 2011, which is prior to when we actually have the genetic tools for it, that was the criteria that we used, was the presence of symptoms and along with the presence of the aerified mites to basically make a call on it. I think what we do now know is it's not 100% foolproof, but it is pretty darn good.
I feel comfortable saying that if you're observing the typical symptoms and the presence of the mites, that it is likely rosette. But if you have the symptoms and you don't have the mites, it doesn't mean it's not rosette, because often the mite populations vary throughout the season. We're receiving samples right now. It's dormant, and we find very, very low number of mites, but we're still receiving positive results for the test. So if you have symptoms plus mites, I would feel comfortable, but the absence of mites, I wouldn't say is the absence of rosette.
Great point. So our next question is, does the virus overwinter live in the soil of a removed infected plant?
In the soil or in the root? In the root, yes. In the soil, no.
In the soil.
Fine roots and real roots, but not soil. I mean, it will not stay infected for very long in a soil environment. If it has made its way into the soil because the roots breaking down or mites dying perhaps under mulch, once outside of a living cell, the Amara virus doesn't do very well. There are some viruses that do. This is not one of those that is stable and able to survive being outside of a living cell.
Dr. James Amrine, he's one of the most important acarologists who's worked on this problem, and he indicates the mites can only survive maybe a day or a few days in the absence of a host. So unless there was rose debris, blooms or leaves laying on the ground, it's not going to survive. If you bury those so that they can start to decompose, that's one way to take care of that, or remove those plant tissues and discard them in the trash. But just laying out in the environment, the mites do not survive very long.
And then therefore the virus will not either.
So going back to the question is, technically the virus does not survive in the soil. It can survive in roots that might be present in that soil.
Correct. As long as the root tissue is still moist and technically alive, even if it's severed from the crown. If you were to cut off, like Jen said, just take the top and not take the whole root ball, those roots are going to survive in certain places within that root ball for quite a period of time. And they, like you said, they may even send up suckers. But as long as that tissue is still at least alive at some level, then it is a potential source of the virus. But then you're going to have to get it back into another plant.
Well, that's a great segue to our next question, which is, let's say there are roots still in the soil. The question is, can the virus be transmitted from old roots to new roots, or is it strictly done by the mite, the transmission?
Okay, at this point, there is no scientific report to show that there is root to root transmission. I think the general thinking is that roses tend not to graft their roots. I can say that there's research ongoing right now to try and see if this is something that's even possible. But at this point, based on the knowledge that we have, root to root transmission is not a given.
We've tried to use the roots are a very good source of virus to work with because they lack the phenolic compounds in the above ground parts, and that makes it a little bit easier to work with the virus. And we've been unable to transmit out of the roots to anything so far. And there was one paper that was able to transmit it to the leaves of certain species of tobacco. We haven't even been able to replicate that. We've done a lot of other work. So if we can't do it using buffers and all kinds of tricks, I doubt that it's happening underground on its own. It doesn't mean it doesn't happen, but it's a very low probability.
Okay, we're going to switch gears here a little bit and talk about some prevention, or there's some questions about prevention. So I'm going to actually give you two questions here at once. Are there tests or planned tests to validate or invalidate claims that hydrogen peroxide is a cure? And can sprays be used to get rid of the disease, or should sprays just be used on new roses?
Okay, I'll take it.
Good luck on peroxide.
Well, I'm not. There are no trials, research-based trials, that have been done that show that hydrogen peroxide or any other chemicals like that have any effect whatsoever. So I would say unless it's been done in a formal study, I probably wouldn't trust that. And then the second question, is there a curative? And no, there does not. Once the plant is infected with a virus, it's basically in its bloodstream. It gets throughout that plant. So just because you cut off a cane, it doesn't mean that you've cured the plant of the virus. And at this point in time, we don't have a viricide for plants that would, make it all better. And so that is why plant removal is the recommended practice. And the purpose of the insecticides is to prevent the infection which occurs when the mites feed on the plant.
The fact that the roots are very good reservoirs for the virus makes that absolutely true because, we've not tested when the virus goes down to the roots and stays in the roots and how long. But if it is in the roots, that means that, one branch, the whole thing doesn't make any difference. There's still some source of virus within an infected plant during some portions of the year within the root system. So, pruning is not even a very good or guaranteed management strategy.
One thing I do want to add is this is an active area of research. So, in terms of viricide, I know people are looking at various products, but at this point, there's nothing yet. And the reason why I bring this up is because it's an active area of research. New information may change the way we recommend what we recommend. So keep that in mind that this is an active area of study and research.
All right, so I have two more questions in regards to spraying plants. Let's see. Question is, so it sounds like horticultural oil before spring growth would be recommended. And then the second part of that is, should you repeat applications of oil as a preventative for mites?
So what I generally recommend is that in an organic situation, you would probably have to use a dormant application during dormancy, and then probably twice a month application throughout the main growing season, keeping in mind that there are some temperature limitations for oils. And so that for certain parts of the country, they may have to take that into effect. You may have to go without during high temperature periods in the summer. For most of our landscapes, for what I recommend is alternating between bifenthrin and horticultural oil. Biweekly would be ideal, so that way you're putting bifenthrin out there about once a month, and it is a systemic. I don't know how sustainable that is, but that is what some of our growers are using at this point. I just wanted to mention, because I don't know if anyone did, that there is a handout link that you can click on that the organizers have put together that has some of these tips and different things that we're talking about. So you don't have to like furiously take notes. It's been summarized in that handout.
So we have a question now. Are there any predatory mites that are effective against the aerified mites?
That's another area of active research by our friends at USDA. We've played around with a little bit. There's potential. Kevin, you know of any good hard data yet?
No hard data yet. There is some observations that have been made, but there has not been any hard data that come out of the studies yet. So that is being looked at. They're trying to mine to see if there are potential predatory mites that would be antagonistic to the aerophyte, Phytococcus fructiferus in this case. But at this point, we don't have any good sound information that would suggest that there is an effective predatory mite.
All right, so now we have a question. Are there varying levels of susceptibility dependent upon the class of roses? For example, hybrid teas, grandifloras, shrubs, etc.
Tom, you want to take a stab at that first?
Well, I'm the one who, my job is to make sure that they're susceptible, and we never did a gradation, and we've never noticed one. I think Mark Wyndham had a more elaborate rating system, and he may have come to some conclusions that Jen might remember, or Kevin. But you know, it's like being a little bit pregnant. If you've got a plant that you can confirm that's infected, even certainly with a PCR test, then it's going to be a reservoir for virus and vector going forward anyway. So from a breeding standpoint, tolerance is not what we're really looking for, because this is an all or nothing kind of circumstance, either resistant to the virus, or at least, if you're going to say tolerant to the virus, at least resistant to the vector then, something to deter their feeding. It's going to be a multi-pronged kind of approach, but tolerance, so far, I don't think it's a good solution anyway.
So let me jump in and echo what Tom said. But to answer the question, are there differences that we see in how
Sure.
fast or the symptoms that appear? The answer to that is yes. Can we classify it by group? You know, I had a student try to do that in terms of symptoms with cultivars. The answer to that is no. You know, maybe, well, I'm just going to stop there. The answer is no.
Yeah, that's the safest thing.
Great. So our next question is it known how long a mite feeds on a rose before the virus is transmitted from the mite to the rose?
I can answer that one based on some old studies that were put together, and I think this was partly some of Jim Amrine's work and some of the folks that work with the mites back, I think, in the 80s or so. So there is at least one paper that said if they took mites from an unhealthy plant and put it on a healthy rose plant, and what they find is symptoms can appear on that inoculated rose, or where they put the quote unquote mites from the dirty plant on. They can see symptoms as early as 8 days after exposing that plant to those mites, and in some cases 200 plus days after exposure to the mite. So it short of it is, we don't truly understand, I think, the latency period. I think in some cases we can get symptoms showing up very quickly. In other cases, you know, a long time later.
We routinely, when we augment ours, Kevin, we will get symptoms in some varieties as early as 1 month later, and that's in spring augmentation. So they're really perfectly set to be transmitting at that point in their season. But that's with a low number of active aerified mites even. So we'll have higher numbers later in the season, but we put plants in, a month later, we start seeing symptoms in some of them.
So what Tom is basically saying, and clarify this for me, Tom, for everybody. Sure. Augmentation means that what Tom did was took branches of infected plants where there's probability of mites there, put it on asymptomatic plants with the hope that the mites would be transferred over or at least walk over and help to transmit that virus into that healthy plant.
That's our standard way. We do not inoculate, we augment. And we also count the number of mites per symptomatic shoot that we put on. So we know we're moving mites and we know how long. I don't think even Jim did the classic studies, Jim Amrine, where you actually put them on for 2 days and then give them an insecticide to kill them. And then he never did one of those kind of studies. But it is relatively quick in feeding period. And it has to do with concentration of the virus and the probability of a mite just having the feeding in the right area on the right plant at the right time. So population densities also of mites.
All right, so our next question is there a recommended disinfectant to use on tools in order to not spread the mites?
I don't believe there's any specific disinfectant. There are so many that are out there. There's not been a trial looking at that to see if one is better than the other. I know that some of the landscapers I work with use wipes. Some of them use where they dip into products as they go along, swapping pruners. And I would say it's more important to occasionally use those disinfectants. And at this point, we don't have data to suggest one is better than the other.
And to keep in mind as well, what Jen is trying to address here is cleaning your pruners from one plant to the next is just good practice. Not just for rosette virus, but there are other viruses and other diseases that could be present. And the question here was asking about the aerophyte mites. Well, if it's an alcohol base or what, it will kill the mites. So we don't have any studies for that, but nonetheless, disinfecting with what commercial products out there is a good practice for just maintaining a healthy plant.
Okay, so now we've got a question this participant says they've been told by a rose breeder that the mite overwinters in the top 1/3 of the plant. So routine winter maintenance of pruning back the problem helps control RD. Is this a fair statement in your opinion?
I sent a number of samples to one of the acarologists that we're working with at Beltsville, and it was collected from various locations on the plants. And where they found the mites in the highest abundance was on the open flowers, particularly on the sepals. However, they could occasionally find them hiding in sheltered spots like leaf axils, so doing good dormant pruning where you're removing 1/3 of that material, it is likely that is what you are removing. However, if you're seeing a lot of leaf material or flowers lower down, it might be good to remove those as well. But it's likely that the 1/3 kind of correlates with where those blooms are being located or that are left over from the growing season.
Also realize one thing. If that plant is infected, by removing 1/3 of the plant, you might be removing the abundance of the mites, but you are not removing the source of the virus, which that plant could be essentially a typhoid Mary type plant, if you think about it.
Very good point. Our next question is there a particular time of year where symptoms are more likely to appear? And is there a time of year where RD is more likely to spread?
We've done augmentation and transmission all year long. It's really more of a question of when you have the populations of the mites and when you have succulent tissue, because those two things go hand in hand for easy transmission. So in about the May, June, and into early July, when we have good fresh growth starting in the spring, we know we have more succulent growth, but if somebody comes in and does some pruning, which is what we do to make the disease worse in our screening block, we can get new flushes of growth. It's the succulence of that growth that makes it easier on the mites to do their transmission work.
And I'm going to jump in here and said I believe it depends on the conditions and in Texas, and Texas is a large state, so we do find different times of the year that it seems to be worse. So typically the top half of North Texas, we tend to see it towards where symptoms start blowing up in the late spring, which suggests that perhaps the feeding or at least the population increase of the mites is happening sometime in early spring. So with mild conditions, surprisingly, in a number of areas, we're also finding rosette popping up in terms of symptoms, popping up in about July and August, which is really hot and it's unexpected. But we suspect it has to do with when we have that flush. So once the flowers are done and it's pruned and there's new flush,
right.
Those things could be the infection court where the mites could feed on and transmit the virus into those plants.
That's absolutely correct. We get a second round after we deadhead some of the stuff, but we're doing that intentionally.
Great. So our next question is, can you explain the correlation between killing the mites and the virus remaining?
Those don't go hand in hand. I mean, if you have an infected plant already and you've got the vector on it and you apply the proper acaricide and you get rid of the mite, you haven't gotten rid of the virus. It's still in the plant till the plant dies.
I think a fun way or simple way of remembering this is an interesting disease system where the virus is the pathogen and the mite is the vector, and so it is conceivable that the mite may not carry the virus. So there are situations where you can see the mite, but there is no disease because there's no virus present. But the mite is capable of carrying this virus. So it's sort of like the virus on its own. If it's in the plant, it's in the plant. It can be acquired by the mite, and the mite is the primary, you can say, carrier for this virus.
Okay, now we have a question about the distance between is there a recommended separation distance from Rosa multiflora?
I don't think anybody's done the
Yes.
studies, but I mean, it depends on wind speeds and all kinds of things. But what's the general recommendation that Jim used to give? Kevin?
I don't remember because I want to say it's like 25 feet or something like that, but it's, from what Mark has done with the various stuff, it's sort of like, for lack of a better term, it's a roll of the dice. It depends on the wind, the environmental conditions, where the mite will balloon to or be blown to or will be deposited. So it's the best answer I think I can give is the further away, the better.
That's symptomatic. That's symptomatic or multiflora they're really talking about anyway.
Right. So it's sort of like, the bigger separation you have, obviously it's better, but it's not a guarantee that you will be free of disease if the mite is present.
Right.
All right, changing directions a little bit. Is bifenthrin organic?
It is not organic.
Now we have a question about, do you know whether mail order nurseries replace infected plants?
They'd have to demonstrate that they came to them infected, I'm assuming.
I would assume that as well.
I think if you're worried about having infected plants that you received in, it's really contacting the State Department of Agriculture and working with them to make those decisions.
Right.
And then let's see, we have one final question, which is, can the disease be spread via rabbits or other wildlife?
That's an
I
interesting
can tell you
question.
a story about a golf course that I used to work with. And the rabbits and wildlife, just them feeding or something like that, aren't transmitting it necessarily. But they did observe, and this is observational, not a research trial, but they had a deer problem. And the deer would come in to where they had a rose garden, and they felt, based on where the deer kind of had a path through the rose garden, that they were spreading the disease. And the thought was that in the process of moving between the roses, I don't know if they were eating the hips or what, but they were getting the mites on them. And then when they left and they went down their little path, they carried it. They felt that it was very associated with the path of those animals. So I would say, yes, it is possible.
There's been
long shown in a research trial.
Right. There's been long anecdotal data that long distance transmission may also be on songbirds who feed in multiflora rows and do a similar thing. And is there hard data? No. But people like to point to the long distance transmission or movement of RRV across the United States and say, well, how the heck did it get from the Rockies to the Mississippi River, other than people digging up infected plant material? They were pointing to songbirds, and there were some, at least early papers, but they weren't very convincing because they didn't have a PCR test to try to test for every single mite.
And hence, I want to point out an earlier suggestion that Jen made to all of you, that realize, if you go to a plot of roses that's infected, don't go to a healthy rose plot after,
right,
because you could be carrying the mites.
On yourself, yep.
On yourself.
Excellent point. So there's a few more questions that have to do with the handouts and the recording of the webinar, so I'll just handle those. There will be a link sent out tomorrow for the handout in a follow-up email, as well as a link to a recording of this webinar. And the webinar will be posted on the Star Roses and Plants website, as well as the Star Roses and Plants YouTube channel. So I believe that was the last of our questions. I want to thank all of our presenters again for providing such excellent information. And I hope everyone who was a participant received the information they needed. There are the handouts available, as well as, again, the webinar will be posted for anyone who didn't get a chance to see it. That link can be sent to people, maybe no other landscapers, growers, master gardeners, etc., that might want to see it. So it will be available.
Thank you.